to activate CAM kinase 2, ERK kinase, phosphatidylinositol 3 kinase, protein kinase A, and or mTOR. All of those kinases can activate the expression of the protein kinase M-zeta messenger RNA. So each of these kinases can relieve protein kinase M-zeta from the initial translational repression. And then the newly translated protein kinase M-zeta is actually active, but co-translational phosphorylation by PDK1 enhances its activity, meaning that as it's being translated, it's phosphorylated by the PDK1, and this enhances the catalytic activity of the protein kinase M-zeta enzyme. This whole process is stabilized by BDNF signaling, which is a mechanism we saw in the previous chapter, and the newly synthesized protein kinase M-zeta is tagged to activated spines. But here's a very interesting trick that protein kinase M-zeta plays. Protein kinase M-zeta, after it's synthesized, phosphorylates a protein known as peptidyl prolyl cis-trans isomerase NIMA interacting 1. Oh boy. So let's simplify things and just call it PIN1. So protein kinase M-zeta phosphorylates a protein known as PIN1. PIN1, in turn, releases the translational block on protein kinase M-zeta messenger RNA. It releases protein kinase M-zeta messenger RNA from translational repression. So in this manner, completing this cycle, protein kinase M-zeta becomes self-perpetuating at stimulated spines. You can see how this cycle would just go on and on and on and on as long as the messenger RNA is being supplied at a constitutive level from the genomic mechanisms and transported out to the dendrites. It's available at spines that have once been activated to keep going and going and going. So every time protein kinase M-zeta is translated at a stimulated spine, and therefore a tagged spine, it activates PIN1, which causes more protein kinase M-zeta messenger RNA to be translated. And there are abundant data available now that demonstrate that protein kinase M-zeta phosphorylates GluA2 heterotetramers to cause them to insert into the membrane and to decrease endocytosis from that membrane. Protein kinase M-zeta also supports the stability of the postsynaptic density 95 structure. In addition, there's abundant evidence that infusion of protein kinase M-zeta potentiates synapses. And inhibition of protein kinase M-zeta disrupts LTP and memory long after their induction, implicating it in maintenance of LTP and memory. So we're going to see some examples of these experiments in the next few slides. I'm going to just kind of quickly show you 4 lines of evidence that support the idea that protein kinase M-zeta is a perpetual signaling mechanism that maintains potentiation. First, when protein kinase M-zeta expression was assessed by Western blots, after tetanic stimulation of hippocampal slices, the amount of protein kinase M-zeta protein was upregulated for hours, in fact, for the duration of the viability of the hippocampal sections. And additional experiments in learning tests revealed that hippocampal protein kinase M-zeta was upregulated for days to weeks. Perhaps most interestingly, the amount of protein kinase M-zeta correlated with performance in learning tests in a variety of paradigms. It's also interesting to note that infusion of protein kinase M-zeta into postsynaptic spines actually potentiated those spines. Looks kind of similar to the effects we previously saw with infusion of BDNF. So this is in the total absence of any stimulation. Just administration of the protein kinase M-zeta itself potentiated the spines. Furthermore, you can see on this graph that co-administration across all of this time of zeta inhibitory protein, or ZIP, actually abolished the protein kinase M-zeta mediated enhancement of the evoked responses in these sections. And I wasn't able to find a very good picture of it, but treatment with protein kinase M-zeta also increased the trafficking of heterotetrameric AMPA receptors into the spine heads by promoting the interaction of the trafficking proteins with NSF and PIC, two transport proteins that interact with the AMPA heterotetramers. Therefore, it appears the protein kinase M-zeta really has a dynamic interaction with these trafficking mechanisms to get those GLUA2 expressing AMPA heterotetramers into the membrane and keep them there. And on the other side, and again, I couldn't find a good picture of this for the slide, but protein kinase M-zeta infusion disrupted endocytosis of GLUA2 containing AMPA receptor heterotetramers, thereby overall increasing the stability of these AMPA receptors in the spine head. And very importantly, protein kinase M-zeta inhibitors, known as zeta inhibitory protein, which we've already seen, or ZIP, and chelerithrine, each reversed LTP maintenance and long-term memory, even when administered hours after the stimulation or learning manipulation. And so in the graph on the left, and unfortunately I didn't have any graphs that could replace these ones from your textbook, we can see that administration of ZIP across the entire time span of this experiment diminished the potentiation produced by tetanic stimulation in comparison with the vehicle-treated sections. And that's when it was administered right from the outset. But what happens when we administer ZIP starting at 2 hours or even starting at 5 hours after the stimulation? We can see in both cases that the administration of ZIP diminishes the potentiation, reverses it so that you do not have long-term maintenance of the potentiation. Clearly, this seems to implicate the protein kinase M-zeta in the long-term maintenance of potentiation after tetanic stimulation. And then in contrast, another protein kinase inhibitor known as staurosporine, and that's not illustrated on these graphs, could disrupt induction of LTP when infused immediately after tetanic stimulation, when, of course, it's going to be able to interfere with all of those different kinases that are implicated in that initial induction. But administration of staurosporine did not disrupt late phase potentiation when it was administered hours after the stimulation. And this makes sense because staurosporine is a non-selective kinase inhibitor that actually doesn't affect protein kinase M-zeta. So accordingly, it seems to be interrupting induction of LTP by blocking the effects of other kinases that are involved in these early stages. However, blockade of those kinases is not relevant for the late phase maintenance of LTP. Overall, this seems to be very strong evidence implicating protein kinase M-zeta in the long-term maintenance of LTP. So it seems like there's a pretty compelling scientific story supporting the role of protein kinase M-zeta in synaptic potentiation, and especially in the long-term maintenance of synaptic potentiation. Everything points toward protein kinase M-zeta playing an essential role as an integrator of long-term maintenance of potentiation and memory. But a challenge came along a few years ago when Volk and colleagues tested protein kinase M-zeta knockout mice for long-term potentiation of Schaefer collateral inputs to CA1 pyramidal cells and for long-term memory in a variety of behavioral tests. So first, Volk and colleagues genotyped their mice and found that the knockout mice clearly did not express any protein kinase M-zeta. The Western blot at the upper left shows that the wild-type mice have protein kinase M-zeta. The heterozygotes that only have one copy of the gene have reduced protein kinase M-zeta, and the knockouts that lack both copies of the gene, of the PKCZ gene, have no protein kinase M-zeta whatsoever. But when Volk tested the mice for LTP, he got an interesting result. In the graph at the lower left, you can see the results for their LTP experiment. They administered theta burst stimulation at the beginning of the experiment in the wild-type mice, shown with the open circles, and in the knockout mice, shown with the closed circles. Do you notice anything wrong? Do you see that the wild-type mice and the PKMZ knockout mice both exhibit perfectly functional late-phase LTP? The knockout mice that hypothetically have no protein kinase M-zeta can still maintain that long-term LTP, that late-stage LTP. And, well, if protein kinase M-zeta is essential for maintenance of LTP, these mice should be impaired in it. But they weren't. So Volk and colleagues investigated further. They tested the effect of ZIP on late-stage LTP in wild-type and knockout mice. Remember that ZIP, the zeta inhibitory protein, inhibits the function of protein kinase M-zeta. So the ZIP, when they tested it, they found that it eliminated LTP in the wild-type mice when it was administered starting an hour after the stimulation. And this is what you would expect, because protein kinase M-zeta is supposed to be responsible for that long-term maintenance. Block the protein with ZIP, and that long-term maintenance goes away. Oh, but administration of ZIP starting an hour after the high-frequency stimulation also eliminated the late LTP in the protein kinase M-zeta knockout mice. Now, how could an inhibitor of protein kinase M-zeta eliminate late LTP in mice that don't have protein kinase M-zeta? Now, there was a puzzle. So Volk and colleagues investigated even further. They tested learning abilities of wild-type and knockout mice. In the left half of this slide, you can see the performance of the mice in the Morris water maze test. You can see that the wild-type and protein kinase M-zeta knockout mice found the platform faster and faster across training trials, and there was no difference between the wild-type and protein kinase M-zeta knockout mice in their performance. And then more importantly, the wild-type and protein kinase M-zeta knockout mice did equally well in searching the target quadrant of the Morris water maze and in crossing over the platform locations in probe trials, even when they were tested after 72 hours. That looks like pretty good evidence of long-term memory, even in the protein kinase M-zeta knockout mice. And in the graphs on the right half of the slide, you can also see that the wild type and protein kinase M-zeta mice do pretty much equally well, certainly no statistically significant differences, in standard tests of both contextual and cued fear conditioning. So since the protein kinase M-zeta knockout mice did just as well as the wild type mice in late LTP maintenance and in learning tests, Volk and colleagues concluded that protein kinase M-zeta must not be the mechanism that underlies late LTP maintenance. And since ZIP impaired late LTP just as well in wild type and protein kinase M-zeta knockout mice, Volk and colleagues reasoned that ZIP must not be as selective for protein kinase M-zeta as everyone thought. So they concluded that some other protein kinase that was affected by ZIP was actually responsible for late LTP in both wild type and protein kinase M-zeta knockout mice. They even published a paper in the journal Nature, in which they reported the results I'm showing you on this slide and the previous slide, and they entitled that paper, "Protein kinase M-zeta is not required for hippocampal synaptic plasticity, learning, and memory." So, I suppose we have to rethink the hypothesis describing that protein kinase M-zeta is a critical memory molecule. But no, not so fast, says Tsoukas and friends. There must be another explanation. Tsoukas and colleagues, 3 years later, replicated Volk's findings and continued to investigate to try to find the mystery protein kinase that was producing these effects. Tsoukas proposed that two hypotheses could actually explain these results. The results of the Volk experiments and of their replication of those experiments. One hypothesis was that protein kinase M-zeta is unnecessary for LTP in memory, meaning that ZIP actually acts on targets other than protein kinase M-zeta, just as Volk had proposed. The other hypothesis was that protein kinase M-zeta is actually necessary for LTP in long-term memory in the wild-type mice, and another molecule, which is also inhibited by ZIP, compensates for the lack of protein kinase M-zeta during LTP in memory in the knockout mice. So they set out to test which of these hypotheses was correct. So I think it's clear that Tsoukas and colleagues favored the hypothesis that protein kinase M-zeta knockout mice compensate with another enduring kinase that's also responsive to ZIP. But now this hypothesis would be hard to test with pharmacological challenges because it turns out that the available drugs all interact with the catalytic domain of the protein kinase C isoforms, and those domains are all very similar. So there's a problem with lack of specificity of the drugs for the individual protein kinase C isoforms. So Tsoukas abandoned the pharmacological approach and examined the effect of an antisense oligonucleotide that was very specific for protein kinase M-zeta messenger RNA. Now, I think I've talked about antisense oligonucleotides before. We have a sequence of messenger RNA that is complementary to the messenger RNA in the tissue. If we apply that sequence of messenger RNA, it will bind to or hybridize with the mRNA in the tissue. And that prevents that messenger RNA from being expressed, from being translated, because you can't translate double-stranded messenger RNA. So they were able to come up with a sequence that was really absolutely specific for the protein kinase M-zeta messenger RNA, not just some pharmacological treatment that approximates interactions with one particular kind of protein over another. So administration of the antisense